My name is Jerry Lin, the Business Development Manager at Omniscient Neurotechnology. Omniscient is a company focused on delivering practical software solutions to connect pioneers in neurosurgery, neurology, neuroscience with subject-specific brain analytics. Our goal was to revolutionize neuroscience with big data. Throughout this webinar, we want to bring you practical insights into the world of connectomics as a brain mapping tool, which can be used for research today and potential clinical uses in the future. Today, we join three speakers. We have Dr. Gloria Yan, who is the Director of Center for Cognition and Mind Science at National Tsinghua University, Taiwan. Dr. Sze-Voon Yee, a neurosurgeon in Hospital Kuala Lumpur, and Dr. Michael Sughrue, the Chief Medical Officer and co-founder of Omniscient. Dr. Yan uses neuroimaging techniques to study neural bases for abstract thinking, neuroplasticities, interactions of language, and other cognitive functions in healthy adults, elderlies, and bilinguals. They also design cognitive training programs to enhance cognitive functions of patients suffer from stroke and brain injuries. They validate the learning effects and neural change with imaging and behavioral experiments and provide data to verify current theories in neuroscience space. Their studies further provide as a reference for neurologists, neurosurgeons, and rehab doctors during treatment. Dr. Yee works at Hospital Kuala Lumpur, the largest public hospital with a dedicated neurosurgery department in Malaysia. The center has 15 neurosurgeons and operates around 2,000 cases a year, with 30% of that in neuro-oncology. Dr. Yee is one of the first neurosurgeons in Malaysia adopting connectomics during neurosurgery, and the centers will be one of the participating sites in Malaysia for glioma connectome project. Dr. Sughrue holds a few different roles. He's a neurosurgeon at Prince of Wales Private Hospital, research director at Charlie Till's Foundation, and co-founder, chief medical officer at Omniscient Neurotechnology. He came to Sydney to train with Dr. Charlie Till's in 2011. He then returned to University of Oklahoma, where he was the director of the Brain Tumor Center. He helped to build the Institute into one of the busiest units in the US. He performs over 3,000 brain tumor operations, making him one of the busiest brain tumor surgeons in America during that period of time. He has published over 250 peer-reviewed scientific publications in medical journals. Joining the dots, he has a vision to move neuroscience beyond evidence-based medicine and onto a data-driven medicines with revolutionary machine learning technologies. Before I hand it over to Dr. Sughrue, there are some housekeeping rules during the session. Please use the Q&A tab highlighted in red for any questions. We'll monitor the questions during the webinar, and there will be some time at the end of the webinar for Q&A. The Q&A sessions may run over on time, but our team members would stick around to answer your questions. Throughout the webinar, you may be presented poll questions. These poll questions would help the speakers to understand about the audience. We'll go ahead and launch the first poll question. Are you currently using structural or and functional connectivity in your current research? Okay, we have 80% with answer yes, and then 20% with answer no. Okay, so with that, I would hand over this to Dr. Sughrue, our first speaker.
Hi, just give me a second here. Hi, everyone. Today I'm going to talk to you about the software solution that we've been working on for a few years now. But I think a better way to think about this is not as a particular software, but as a different way of doing research. And so I called this lecture the Infinitome concept. And where the Infinitome concept comes from is the observation that the brain is extremely complicated. And as such, diseases that happen in the brain are extremely complicated. The brain has billions of connections. It has very complex communication patterns. And as a result, when we start to look at patients with the multiple forms of information that are available in clinical spaces, we notice that these patients have diverse symptom profiles. And given that the other numerous ways that patients are different from each other, from medical history to other life experiences, et cetera, it really highlights the fact that we're dealing with an extremely high dimensional and data intensive problem. And up until machine learning was a tool that could potentially tackle the complexity of this problem, it was really just something that we put our hands up and said, well, you know, we'll do the best we can. But what I want to try to convince everyone is that the tools are beginning to become available to us to be able to tackle extremely high dimensional problems, and to do so working in the complex and highly regulated medical legal environment. So given that about 80% of you said that you are already using some of these technologies, let's just briefly touch on what we mean by connectomics. So connectomics comes from a portmanteau of the word genomics and connections. So genomics are big data analytic bioinformatic approaches to the genome, and connectomics thus is a big data analytic approach to the connections, both structural and functional, in the brain. And so again, what people in many cases, especially clinicians, are less aware of is that we have reasonably detailed maps of at least the macro scale connectome. What we don't have is right now is a full understanding of the significance of all of these connections, but it really shows that with high resolution functional and structural imaging that's becoming increasingly widely available, and utilizing especially big data analytic techniques, there's a lot we can begin to learn about what's wrong with our patients, and more importantly, how do we leverage into actionable insights. So I became aware of this several years ago when I really, as a neurosurgeon, just wanted to have better maps of the brain to do better brain surgery. So you can see on the GIF on the right that's running on a repeat loop, this is a modern language map, and it comes from a lot of work by many people at many sites to try to update the Broca-Wernicke model to explain more of the both basic science and clinical data. But it's not enough to make pretty pictures. And again, really being able to mine the data with massive scale machine learning, a term called federated machine learning, which solves some massive problems in collaborating across sites, and integrating into the systems that we acquired data, really is going to be essential to begin to gain additional insights into the connectome. But more importantly, it's become increasingly clear that no individual site running any study of any reasonable amount of effort is likely to see enough patients with most of the diseases with specific symptom profiles to really be able to handle this on their own. And we need to learn to work together. And the Infinitome concept is a concept of utilizing technology to eliminate some of the problems that are involved in doing big, hairy, audacious studies. And in doing so, it's important to be able to have appropriate feature labels and good clinical metadata to begin to feed the machine learning models. And again, I'll talk about some simple tools that we've created in this workflow in that regard. But ultimately, what we're trying to do is to utilize engineering solutions, data analytic solutions, to really be able to harness the power of the machine learning techniques that have become widely available in the last 10 years. Computer science, and particularly data science, has undergone a massive scale revolution. And it wasn't because people invented convolutional neural nets or other things. Those have been around for many years. It's the computing solutions that make this actually practical. Now, part of this also is that if we gain insights without having good visualization techniques to view these insights and to leverage this into something that clinicians can understand. It's very, very interesting observations that will often sit in the literature and not help patients. And so one of our angles and something that we've tried to really build into the DNA of our company is that we're not a data mining company. We're a company that makes doctors smarter by ultimately facilitating large-scale research projects that can be pipelined into actionable insights that people can use in the real world. And my former chairman at Oklahoma, Tim Mapstone, mentioned this to me one time, and it really stuck with me. And the way to think about this is that we have one of the largest scientific neuroscience experiments imaginable going on in multiple places all the time, and we're not utilizing it, which is hospitals. We have lots of sick people coming with neurologic diseases, and we really don't study very many of them, because right now we think about the Research Institute as being a different place with a different set of protocols, and we really haven't tried to figure out how do we study the patients where they are. And again, in hospitals are generating large amounts of data that mostly doesn't really go utilized. And so we have this data stream, and a very small bit of it goes over to the Research Institute, and then we wonder why our progress isn't as fast as it could be. Well, there were some challenges with doing this 10 years ago, but I'd like to argue that this is not as much of a challenge as it used to be. It is possible, or at least conceivable with the right infrastructure, that we can mine a much larger percentage of the data coming through the hospital and learn at a much faster rate than we're doing. So Infinitome basically builds around connectomics, and really we aim to have connectomics that we could actually utilize in a real world setting. So while the Human Connectome Project did some amazing quality imaging, it's very hard right now without a major upgrade in MRI speed and acquisition parameters for us to envision doing that kind of imaging in a clinical setting. In fact, it's very difficult to add even 15 to 20 minutes onto an MRI without this being a cost detriment to most hospitals and really a non-starter. So we really work to try to focus on creating noise reduction, cleaning, and processing tools that can work with clinically applicable MRIs, and try to get as much data out of that as we could, acknowledging that there would be some signal loss, but that it would be pretty good given what we could accomplish, especially if we protocoled and acted this on the front end with partner institutions. And once you acquire this data on a large scale, we utilize very large scale processing tools to acquire similar to HCP type pipelines as far as data acquisition. There you look and see a large adjacency matrix. And what we're really trying to do, though, is to collect a lot of data out of this imaging and supplement it with neuropsychological and medical record type data in a way where this all goes to a single place in the cloud, allowing eventually for people to speed up the process of sorting and finding patients of interest. So again, the thought process is, instead of going and signing up 20 patients that fit your inclusion criteria, you sign up everyone and go back and find them using a tool like we see there. And allowing people to launch massive scale machine learning tools to try multiple different models in parallel to find best fits and to fail quickly, which has become a really important insight coming from the computer science world, which is use agile workflows, fail quickly, and then develop promising hypotheses so that you, again, expedite and speed up solutions to problems. And ultimately, because we're working with cloud, we have the ability to get sites in different centers and even different countries to work together in ways that preserve and obey the numerous data regulations around HIPAA, GDPR, et cetera, but still be able to harness the power of machine learning in ways that really would be very difficult to do without having a very, very large grant. So digging into that a little bit deeper, fMRI or DTI are not new. And while we utilize fMRI, particularly resting state fMRI and diffusion tensor imaging, we're not the only ones doing this. And again, these are not new technologies in any way, shape, or form. If you look at this, these have been around for about 30 years. And last time I looked on PubMed, there was about 550 peer-reviewed publications. And probably everyone on this, or almost everyone on this video, has heard and knows a fair bit about these topics. But then it raises the question of, if this has been around for 30 years, and in general, the validity of at least some aspects of these techniques are not really particularly controversial anymore, why aren't we doing this on a regular basis? There are a lot of tests that we do that have far less sensitivity, specificity, far less understanding of the pros and cons, and far less potential than fMRI and DTI. And the ultimate solution, that problems that I found when I began trying to do this at an institution where I didn't have expertise around every corner and had to do a lot of this myself, is that the software solutions are quite limited. And even at places where there is significant expertise, it's logistically painful. I mean, again, if you have a surgery tomorrow and you want to get some advanced imaging on somebody, that's not probably going to happen, because unless you have a PhD that's willing to drop what they're doing on a moment's notice and live into clinical workflows. And as a result, what we have are solutions that are not very scalable, even inside world-class institutions. So it becomes a self-fulfilling loop where we as doctors don't get it, so we don't learn what to do with it. And because we don't learn what to do with it, we don't bring new insights into it. And again, the validity and utility of some of these techniques is really just not very well understood. And what we then get is people making amazing looking pictures like this that serve as great desktop fodder, but ultimately really don't tell us what we need as physicians, even though we know that the data is probably in there somewhere. And so really being able to go in and annotate the connectome, link it to things that we care about as far as clinical, symptomatic, and other neurological benefits is really the difference between having a nice poster in your office and something that actually impacts the lives of individuals. Going a little deeper, this is my wife with Grammy Award winner Brian McKnight. And if we think about our studies and trials, and why many trials that seem to have promising biological efficacy for drugs or other interventions often fail or are not replicable. Well, it raises the question of, should these two people be controls for each other when they're obviously different? And not just from a physical appearance. Even if they were identical twins, we know that there are differences in their connectome, their genome, and we're randomizing them against each other. And so ultimately, when we run a trial like this, we're assuming when we randomize people against other people that we're really studying this, when in reality, given this, we're really studying this. And so the only things that really seem to break through these are things that are either so overwhelmingly beneficial, such as removing a clot in a ischemic stroke where it's obviously it seems likely to work, or something where we just happen to get fortunate that we have a big enough study that averages out the heterogeneity. Many times we probably have neglected or had failed studies of chemicals that might work in the right patient cohort. So in reality, what we should be thinking about is, how do we utilize technology to randomize people against themselves or against a virtual version of themselves, so that we can make cohorts that look more like this: smaller, more efficient, more agile, and more specific. So the first step towards at least handling the connectome, obviously other people have been handling the genomic heterogeneity and working towards this in different ways. But one of the thoughts of how to handle the connectomic heterogeneity, which is more important for other diseases, some diseases than others, is that how do we make processing large-scale data simple? And one of the biggest challenges that we found, particularly when I started looking at my own patients who all had tumors in the brain, is atlasing. So what an atlas technically does is, let's say I'm trying to assign every region in the brain to an area, or what's called the parcellation. And there's a lot of different ways to do it, but most of them essentially do some format where they take a standardized atlas where all the coordinates in this area get assigned to that parcellation, and the ones in this area get assigned to this one. They could do it volume or surface or whatever, but again, it makes an assumption that the brain hasn't changed shape in any major way. And in fact, many of the software programs will crash if you try to pass in an abnormally structured brain into there. And so ultimately, what we started working to is, can we find a better and faster and more scalable way to assign a parcellation to brains regardless of their shape? So what we created was a machine learning tool that would look at the tractography, in this case, not DTI, it's the next generation CSD, which handles crossing fibers a little bit better. We trained it what every single area, every single parcellation, for example, Broca's area, every single area's voxel look like, what a voxel of each one of these areas look like based on who they were communicating with, what their connections were, such that we could make an atlas that was more adjusted and personalized to that individual. And once you have an atlas, once you've assigned everything, you can then compare every area with every other area as far as the bold signal in an fMRI. So you can say how much is this area talking to this area, using different statistical methodologies, and create this sort of matrix. And what you're looking at here is each square is a comparison of how much one area correlates with another area. But this is useful, but what's more useful is actually utilizing machine learning techniques to tell you what's not normal on here, or at least what's an outlier. And we'll talk about that a little bit. So again, once you have an atlas, there's a lot of really interesting things you can do about it. Most notably, you can bring knowledge about the brain, anatomical knowledge, back into large data. So for example, if an area maps to 44, well, it's not on the left side. We can't be certain it's language in that individual, but it's a better candidate for language than, say something in the parietal lobe, or even something up here. So it lets us put some structure on our data. And what it also allows us to do is to do things like taking a very complex set of waveforms, which is the bold signal at these 2 points, comparing them, and acquiring a correlation. What you're seeing here is a giant correlation matrix, where the red squares are areas that are highly correlated with each other, and often they're forming networks. And the blue areas are areas where 2 areas are anti-correlated, they're opposite each other. So what you can see here on this axis is, this is a list of every single area of the brain that we map, and this is the same list. And what you're looking at is about 143,000 comparisons. So these are very large and high dimensional data sets. Again, once you have a nice pipeline, if you have it all in loose code and it's not engineered correctly, and it's going to run slowly, and it's going to require expertise. And one of the biggest challenges is actually just getting things out of packs and putting it onto the server. So we've created some very unique solutions to this. Most importantly, that it de-identifies and defaces the data, so that we actually don't store anything identifiable in our cloud. We try to make it almost impossible. I'm sure someone can hack the DICOM header to put that in there, but in general, unless you're trying to sneak around this, you won't get PII into our cloud. It doesn't accept it. And again, what this aims to do is to make ordering an fMRI essentially like ordering a sodium or some other basic lab test, where this isn't really something that we're requiring manual work to process. But once you have things into the cloud and you're dealing with high dimensional data, it really is important that you throw at it tools that are appropriate to the task. And some of the most important thing in machine learning aren't the algorithms. In fact, the algorithms, if you're familiar with how to run, for example, an SVM, it's a couple lines of code and some structuring around that to get the data in the form that it will understand that. But it's not that hard. That isn't the challenge. The challenge is that some of these models really need a lot of data, particularly the deeper learning models, and they need a lot of time. And so if you're going to get this done in a reasonable time period, it's best to be able to process them at scale and to really be able to take advantage of parallel processing. And so in that regard, we built a machine learning workbench that builds into our cloud that is capable of processing at least 1 million scans at once. Probably there's no limit up to what AWS will provide for us. But again, there's not enough data in the world right now that I'm aware of to actually meet that demand, but it is possible to do that. Now, having loose scans without knowing who the patients are and what was wrong with them aren't only so useful. I mean, you can use them for analyzing and refining methods and determining noise reduction and things like this. But if you really want to make a difference in people's lives, you have to know what was wrong with the person so that you can learn what's wrong with the brain related to this. So we created a few tools designed to expedite this. And so a lot of challenges with regards to running trials and really cost is paying for people to look through medical records or collect data prospectively in some format and manually enter them into a database. This really seems ill-advised given the technology that's available for other companies use to sell us advertising. And so we utilize some of those technologies such as natural language processing that can look at medical records in English and Chinese presently and eliminate things that we don't need, to read the medical record and extract medical terms and to know where it got it from. It knows how to read that this is an H&P and where to get different information from. And more importantly, knows how to correctly read this. The reason it knows how to do this is that natural language processing tools are rule engines, and the rule engine is based on me. It reads it like I would want to read it because I helped build the rules with the developers, and I've written a lot of papers on medical records if you've ever looked at my CV. So I think it's a pretty good tool for, most importantly, identifying who the patient is so that we don't have to go in and find this retrospectively, particularly if we're collecting data at large scale. In addition, for anyone who's done neuropsychological testing, we know that while it's ideal to have a 5-hour neuropsychological battery, most patients cannot get this test. And even if they could, neuropsychologists are usually, in most institutions I've ever been at, are overwhelmed with work. And that's assuming you have anyone who's very good. And so we really work towards making neuropsychological tests that are modified versions of known tests that have good track records, but more importantly, are simple to do by people who are non-experts. And we created a tool that can basically not only collect the data, but to do things like take the voice to text and determine, is this a correct answer to the question? And right now, when you start talking about looking at aphasias scoring, we're performing at 92% so far. And we think that this has a lot more potential than going for the perfect solution, which is just not scalable and achievable at most institutions. So once you have all the data in a single place and you can identify, I want to find a new onset glioma in the right frontal lobe with these symptoms, or an Alzheimer's patient who has these symptoms, who's on this medicine, then and again, we're working to create tools to query such an engine. And this is actually a fairly sophisticated thing to do, because even though this looks simple, it has to basically link to querying algorithms. And this is a lot more complicated than you would think. And with the eventual goal, though, of, again, getting this into large-scale machine learning, cloud-based training models, where we take the programming and all the other skill sets necessary to do this research currently and make it available to everyone, regardless of whether you have someone who's a Python programmer on your staff. And again, putting the data into a single location, having it processed through large-scale processing cores is all great. But one of the challenges is that, let's say you have institutions in different countries. And let's say this is, for example, China, where it's illegal to send medical data out of the country, and the United States, where it's at least highly discouraged and quite challenging to work through all of the legal contracts with different institutions. And currently, what you would have to do is find some kind of way to work together or look at this data. It just is, and if nothing else, it involves passing around large amounts of DICOMs and medical records and annotating this in a spreadsheet, and sometimes it's wrong. This is really not the best way to do this. And ultimately, there's a cost associated with this, and a cost associated with just hiring enough people to do all this work, supervising them, auditing, all of this. But again, when you go towards cloud solutions to this kind of issue, it makes working between groups in different countries a lot easier, because you don't have to have the data. All you have to do is send your algorithm out to query it in a methodology called federated machine learning. And so federated machine learning does an analytics brings back the weights of a machine learning model and combines this at the mothership without having physical possession of the data. And again, this massively simplifies a lot of the concerns about data selling, data ownership, privacy, all of this stuff. And more importantly, all of these tools can drastically reduce the cost of having a multi-center study that really begins to do things that are high quality, high impact work without having to get a $5 million grant, which we all know are very, very competitive and difficult to get, even if you're an excellent group. So what I've hoped I convince you is that if it is possible now with the right mentality to begin to turn your institution into a research machine and a big data machine. And we have the tools to really help you do that. And again, the Infinitome concept is something that we're always working to evolve to make this better and really more simple, because we really think that in the long term, the neuroscience should be done by neuroscientists, and this should not be based on who is actually the best programmer or has the best programs. So thanks, everyone.
Okay, just to, as a reminder, if you have any questions for Dr. Sughrue, please use the Q&A tab. Next up, we have our next speaker, Dr. Gloria Yan from Tsinghua University, Taiwan.
Hello, everyone. Let me share my slides with you. So today I'm going to introduce everyone introduce to everyone what we have done using this great platform to analyze the AD and MCI data that we have collected with collaboration with Tzu Buddhist Hospital and also the aging centers nearby. So my lab and also my center, over the years, we have done all kinds of analysis of connectivity, including DTI tractography, functional connectivity, independent component analysis, voxel-based morphometry, you name it. But over the years, we have found that through all those approaches before, what we have got are the kind of loose correlations or a general kind of phenomena. So using this platform, we are thinking that we will be able to get a new insight, particularly from the viewpoint of hub. So as you can see that from my topic today, we would like to use hub opacity, hub opathy in Alzheimer's disease. So this will be a new approach that for us to examine the longitudinal change in MCI and Alzheimer's disease. So hub opathy actually implies that the areas which are highly connected to other areas in the brain are targeted by neurodegenerative disease. So this means that we should not just look at a particular small phenomenon such as cortical thinning or a correlation of a particular region with other areas. What we should think from a different perspective is that we should pick out the important hubs of cognition in the brain. And by looking at the change in those hubs, we will be able to get new insights of how the disease or neurodegeneration develops. So we apply the hub concept to find out if there's any difference in degeneration pattern of hubs at different stages of the brain disease. So in this study, we compared the MCI, mild cognitive impairment, and Alzheimer's disease. So we had 33 AD patients, 85 MCI patients, and 93 healthy controls. So using this platform, the platform automatically does all the diffusion tractography preprocessing for you. So it will reslice and also did the motion correction and also did all the scalp stripping, eddy current correction, all of that. So which is very much like what the previous speaker already mentioned to you, that you don't need to train your grad students to do all those preprocessing anymore because the platform automatically does it for you. So after the platform does all the preprocessing of the DTI images, it also did the creation of the personalized brain atlas using machine learning. So basically the platform uses the Human Connectome Project's MMPI atlas and in the MNI space. So it was using that as a way to postulate all the brain regions in the brain. And based on that, everybody was postulated using this similar atlas and compared it based on the connectivity results, not just by the traditional normalization or segmentation, which has been criticized in a lot of literature. Because as you know, when the disease progresses, the brain area, the cortexes, surely will go through some shape change or thinning. So it is really not right to just use the conventional normalization approach to be applied to everybody. So instead, what this platform does is that it picks out the structural connectivity of everyone and use that connectivity and postulate it based on the HCP MMPI atlas. And again, average that, use the whole thing as a new atlas as our basis for comparison. So after this machine learning postulation, and we first try to look at the fractional anisotropy of everybody. We hope that perhaps the FA values in everybody as an average could show us some particular patterns. So we first pick out the areas, the average that shows the FA values are particularly low. For example, the FA under 0.05. So those regions showing FA less than 0.05 are the voxels that show very poor structural integrity. So we first look at those. And here in the diagram, you can see that the orange are the MCI patients and the blue are the Alzheimer's disease patients. So what we see here is that we see a very similar trend. So all the voxels are around this range between the here and there. And also the MCI showed a similar trend. So by looking at that, okay, so the voxels with particularly poor structural integrity, and the trend is similar. So we did not see much. But what about the tracks that are showing very high, very good FA values, which shows that the structural integrity is still intact. And again, we see very similar trend. And sometimes the MCI patients actually similar to the poor FA values, the MCI patients, the values are higher, but still the voxel, the average voxel are still cluttered around the same area. So here, what we see here is that if we just look at the total average FA values in MCI and AD patients, we just see a substantial overlap, and the trend is not so obvious. So based on what we saw just through the first attempt, we saw that the loss of structural connectivity features and might be more important than the overall FADC decrease. So probably we should focus more on particular regions in the brain instead of the overall FA. And also we look at the graph matrix such as the total sum of edges and global efficiency, which I'm going to explain to you. So by looking at those, we are hoping that the graph matrix will give us better insights. So we first look at the sum of edges in both AD and NCI groups. So the sum of edges is the sum of edges calculated in each individual's connectivity matrix. So the sum of edges is calculated based on the count of the edges. And the count of edges is calculated based on all the links connected to the pairs of nodes. So by looking at this, we see that although there's a significant difference between the two groups, we can still see that the sum of edges, the pattern is still very similar. And the range is also very similar between 0 to 20. And so we are thinking maybe, how about we look at the efficiency? So the efficiency is calculated based on the inverse multiplicative inverse of the shortest path distance between pairs of nodes. And we obtained the average of all the like voxels, all the nodes in the brain. So by looking at this, we notice that AD and MCI patients, again, the trend is still very similar, and the difference is significant here. So what we are seeing here is that perhaps if we just look at even the graph matrices and also the global FA values, the group measures might fail to explain what's really going on in each individual. And of course, we know that in all kinds of like neurodegenerative disease, there is a pattern, a general pattern, but everybody would surely exhibit individual difference. So here we see that perhaps we should use a different measure to reflect this difference. So we selected page rank centrality as a measure, try to compare the MCI with healthy control and also MCI with AD patient. So the way we calculate the page rank centrality is like this. We first listed the page rank centrality for each brain region in an individual ranked order from 1 to 379. And then we selected the median rank for each supracortical region in the healthy subjects. Then we set this as a default listing order. And based on that, we create the rank of a specific individual brain region. And also we compare the median rank of this region in the other healthy controls. So with this, we will be able to compare the relative centrality of a specific area within an individual person's connectome compared to other areas. Okay, so here from this graph, you can see that if we use the page rank score as a y-axis and we try to compare the three groups' performance, their listing, their comparison result with the default page ranking, we notice that the blue one and the red one are the blue one is MCI, the red one is AD. So the blue one and the red ones, there's a strong overlap. However, for the healthy control, the green line shows that in the top ranked, those top ranked hubs and the behavior is totally different. So here we notice that perhaps during this kind of neurodegeneration, the major hubs that shows very high page rank centrality in the healthy controls are quite resilient. So here we try to compare the healthy control with the MCI. So as you can see that the top ranked, the hub are listed here, the brainstem, left cerebellum, and the thalamus, those regions. But if we pick out the subregions here, here on the right, we show you a brain in which the MCI shows very different pattern from the healthy control. So the MCI patients here shows that here the blue one is a thalamus, And so the thalamus and the BA, and also the left auditory area and the BA9 here, a medial frontal, are very different from the healthy control. So this means that during this MCI stage, when people start to show the amnesic signs and also some deterioration of certain behavior, the hubs that display the change actually are shown in thalamus, the auditory region, the somatosensory area, and the anterior part of the prefrontal cortex. So this is quite interesting because in the past, a lot of studies hypothesize that all the change starts from the hippocampus or the entorhinal cortex. So a lot of analysis has been devoted to the analysis of hippocampus, the cortical thinning, and the correlations of cortical thinning or voxel-based morphometry change, all centered around the temporal region. However, if we just look at the hub itself, we notice that the frontal hub are actually quite affected, and then the somatosensory hub is also very affected. But however, this doesn't mean that the pathological change or the tau pathology or amyloid beta pathology happens already in the frontal regions. Probably the tau pathology and amyloid pathology, they appear in the temporal lobe. However, they break the connection to the frontal regions. So that's why the hub in the frontal areas are affected. And here we compare the healthy elders with the Alzheimer's disease patients. We notice that in addition to the somatosensory area, auditory area, thalamus, and we see more frontal degeneration, particularly the anterior part of the PFC, and also the precentral gyrus, which is the primary motor area. And the yellow one is a language area. So this means that when people progress to the AD stage, the frontal regions, the language network, the executive network, the executive function network, and also the motor network are all very affected, are impacted by the pathologies happening for the Alzheimer's disease. So after comparing the healthy elders with the MCI and AD group, so we were trying to figure out, here is a question, that what would be the brain signature that shows you the conversion from MCI to AD. So in the past, we all believe that the further degeneration from the temporal and prefrontal cortex and even the parietal areas could be certain indices, because a lot of previous studies have shown that there are a lot of correlations of the cortical thinning or voxel-based morphometry change in those different areas with different neuropsych scores. And so based on this page rank centrality measure, we notice that actually the entorhinal cortex, the parietal occipital juncture, the sulcus, and the precentral gyrus, and also the very anterior part of PFC, those areas are the hubs that show you there's the signs for the conversion from MCI to AD. So what is important here is that we can see that in the past, without looking at this connectivity change, we were only thinking about the neurodegenerative disease in terms of like parts and parts, little bits of parts. They are correlated with different cognitive functions. But however, using this hub-opathy view, we are looking at it from a different way. The different perspective that we are showing you here is that actually when people start to really experience the the important symptoms, the executive function network already shows you this serious breakdown. So that's how this really lead to the big change. So, and again, our hapopathy analysis prove that. So here again, we can display that our conclusion is from the normal to NCI stage, when people start to show amnesic signs, the change, the hap change actually occurs in the thalamus, in the somatosensory areas, and also the auditory region, and only a little part of prefrontal area. But however, when people start to change and degenerate into this AD stage, the more and more like prefrontal areas are involved, including the areas processing language and working memory and the planning for future actions. So those areas are really important for people to carry out more complicated, higher order cognitive functions. But however, if we want to find out the neural signature for conversion from MCI to AD, we notice that the parietal occipital sulcus, the motor network, and also the working memory attention network, and those areas are actually the hub that shows you the conversion. So, so we think that this approach is really, really interesting and have provided us like whole different view to look at the brain change, especially in dementia. So if you already are a member of a dementia center who own a lot of like the longitudinal data, it would be nice for you to compare those change to see whether it also exhibit this similar pattern. And it would also be more ideal that using this platform that can like keep all your data processed in a good pipeline in a secure place. Different multiple centers, we contribute our data, put that in this platform. And perhaps with more data, we will be able to see more interesting phenomenon. Not just this, because usually for machine learning, the more the better. And in addition to that, if you are interested in neuropsychological change or even some psychiatric symptoms, it would be nice for you to perhaps discuss with us or the Infinitome group to initiate a different study focusing on a particular symptom or psychiatric disorder or phenomenon associated with this kind of pathological change. So here is what I have to say for today. And if you have questions, please feel free to email me or contact the Infinitome group. Okay.
Thank you, Dr. Yen. That was a very interesting finding. So hopefully one day with the collaborations with other institutes, you know, we can find the biomarker for Alzheimer, you know, fairly soon.
Okay, thank you.
Thank you, Dr. Yen. If you have any questions, you can either send it to Dr. Yen's email or you can just drop, use the Q&A box below. We can answer your questions at the end. Next up, we have Dr. Yee from Hospital Kuala Lumpur.
Hi, hello everyone. Can you hear me?
Yes, we can, and I can see your screen. All good.
All right, okay. So very good evening to everybody. So today I'm just going to talk about a case, one of the first cases that I've actually incorporated in my current practice right now. So it's actually a recurrent low-grade glioma case, as you can see. So I'll jump straight right into the topic today. So let's have a look at this case. So I have a 36-year-old female here. She's a right-handed. She's unemployed, and she actually presented to us in 2017 with seizures. So we started off with tonic-clonic seizures, then followed by generalized tonic-clonic as well. So we actually scheduled for surgery. So excision was done in 2017. Post-operatively, she actually developed right hemiparesis, which persisted for about 6 months before she regained a full control of her right sided of her arm and also her leg as well. However, her ankle remained pelagic until today. So HPA at the point of time showed diffuse astrocytoma, grade 2. So 3 years down the road, we followed her up closely, and in 2019, we showed her repeated MRI actually showed there's a tumor recurrence, and her seizures has increased as well. So let's have a look. As you can see here, the preoperative image and the postoperative image 2019, the tumor has actually grown almost similar as it was before in 2017. So at this point of stage, definitely we would have recommended resurgery for her. However, at that point in time, she was a bit reluctant. So we managed to convince her this year. So she have agreed to go for surgery. So at this point of time, I'm pretty sure that all of us would think, how do we approach this, right? We have thought, and then all of us would have known that the best way to do this, a recurring case, would be a multimodality approach to it. First, to enhance maximal safe functional resection. So as what we have always do, MRI as a preoperative imaging with DTI, awake craniotomy if that is feasible, intraoperative ultrasound, IGS, and even intraoperative CT or MRI, which could be used as to assess the degree of excision. And last but not least, our intraoperative brain mapping, motor mapping, cortical and subcortically. So in February, so this year, I came across connectomics, and what was shown there actually caught my attention a lot. So I went, did some research, got in contact, and also I did some of the literature study, and I found that actually connectome has been used, and most of the time in 2016, as shown here, is mainly just for preoperative brain mapping. However, this year, as in published in January itself, there actually publication which shown that planning has been made as a preoperative tool. So let's see. So I've decided to incorporate connectomics into this patient. So before that, let's see. I have a few questions in mind actually when I'm looking at the case. So can the software itself accurately locate the cortical spinal tract itself and also the functioning areas of the motor and the premotor areas? So also, did the motor cortex and the cortical spinal tract actually reorganize itself? If yes, where are these areas now? So let's have a look at the images. So as you can see here, the pre-op connectomic image actually show that on the pathological side, the tracts are all being displayed, all being splayed laterally and also posteriorly as well, as compared to the normal side. Okay, you can see the tracts here on the normal side is no longer seen. However, at the pathological side, you can see still some tracts are being splayed. And a lot of them are actually originating from the lateral margin of the tumor. So surgery was done. So how I did it was, we did it under general anesthesia. I did not use awake in this case because the patient was a bit anxious and she's not so keen for it. So yeah, so we did it under sleep. So intraoperative ultrasound was first used to locate the tumor and also to find the border between the normal brain and the tumor itself. So what I found when I was doing my mapping, I found that at the lateral margin and also at the posterior margin is where I found a positive irritation. Whereas as mentioned, the lateral side corresponded to the thenar region and the posterior part corresponded to the quadriceps region. So only the medial portion has negative response, and that was my entry point as shown here. And in the preoperative imaging, the medial part of it is devoid of any tracts as well. So resection was done, and then until it was about, when I resected all the way until about 5 mm away from the cortical spinal tract. So I thought I did a right job. So however, what I found post-operatively was immediately post-op, she was pelagic. There was no movement on the right side of the body. And yeah, but that made me think that it could be just apraxia. So we'll see. So true enough, 24 hours, she regained power of a lower limb. However, her upper limb remained pelagic. So I started thinking, what went wrong? So could connectomics actually show me that I've actually over-resected? So we did a post-op MR for her. As you can see here, the tracks are pretty much similar to the pre-op. And now you can see a lot of the tracks right now has already opened up as compared to the pre-operative where they are being compressed laterally. Also, you can see some of the tracks is no longer staying at the medial portion and at the lateral portion, and a lot of them, some of them are even started to come from the medial portion already. Okay, so let's have a look at the pre and post-op. As you can see here, the pre-op, as I mentioned earlier, most of the tracks are coming from the lateral side of it. And then the post-op ones, some of it are already coming from the medial portion. Also, you can see some of the tracks here, the lower portion of it, you see some of the green fibers are no longer seen. So I thought, could this be the fibers where the upper limbs are? However, my mapping only showed me that the irritation at the posterior margin of the tumor are only corresponded to the quadriceps, none on the upper limb. Most of the upper limb irritation came from the lateral margin and a bit of mainly at the lateral margin, yep. So that made me think again, is there something wrong? Did I resected some of the cortical areas? So I have a look at the pastillation as well. As you can see, the pre-operative pastillations and the post-operative pastillations, the areas have shifted as well. However, the numbers seems pretty much similar as well. And you can see right now, post-operatively, a lot of this area at the medial and the posterior and inferior margin has already been filled up with areas with functional motor areas already. So yeah, so I thought it could be something to do with the areas that are talking to each other. So I've dived into the table, the connectomic tables, and this is what we found. This area in the left putamen and the right supplemental motor area seems to be more hyperactive. As you can see here, evidenced by the connectomics table, where the red voxels here are showing slightly more active compared to the normal areas which are shown as black and white. So that made me think again, could this area is responsible for her temporary praxia? I really can't say for sure, but the data is showing me something. So it could be true. So yeah, so just a final update on the patient progresses. So at day 14, she actually did regain control of her upper limb, but it started only at the distal with her fingers and it progressed proximally. So at day 30 itself, she had full power of grip strength, elbow and wrist. However, shoulder remained at 3. One thing, I still find that she cannot have fine motor of her fingers yet. And the lower limb power has improved as well, only at the hip. The knee is at 3, the ankle remained 0. So yeah, so back to the question that I asked myself just earlier. So did the software accurately locate the corticospinal tract and functioning motor areas? I would say yes. And did the motor cortex and corticospinal reorganize itself? It's yes as well. And they have, as mentioned earlier, as shown in the scan earlier, and most of them has actually relocated themselves posteriorly already. So yeah, from this case, I've learned that the connectomics actually is pretty accurate. And I would say it's almost on par with the subcortical mapping. And then as well as the planning, preoperative planning is pretty accurate as well as the entry point. And this could be registered earlier and also to identify where is the entry point. And subsequently, during the surgery itself, we can use it as a guide together with our intraoperative neuromonitoring to enhance the safety again. So I also learned that, especially when we are very near the corticospinal tract, we have to be extremely careful or else a permanent damage could be done to those tracts. And as well as I mentioned, despite an intact corticospinal tract, plegia could still happen. So importantly is if there is no corticospinal tract and cortical damage, I assume that recovery should be able to occur in this patient. So yep. So I guess a take-home message for today for this case is, regardless for this kind of cases, I think a multimodality approach remain the best way to tackle this kind of gliomas. So, and connectomics is, I would say, is definitely useful in our preoperative planning as well. And also intraoperatively, especially the subcortical region, when we are resecting it, I think this will be a very, very important guide as well. And then the shift in subcortical regions, I think it's pretty minimal as compared to the cortical areas. So I think it will pretty accurate. It will serve as an extra guide on top of our intraoperative neuromonitoring. So yeah, and here I'm going to finish off my slide, and there's just a special acknowledgement to my team, my consultant, Sughrue, and my neurology team, who has been extremely helpful when we are first doing this sequence and also deciphering the tracks and also the pastillation as well. Dr. Rose, Dr. Fiza, Dr. Tay, thank you. So with this, I will end my presentation.
Thank you, Dr. Yee. That was really excellent presentation. I'd like to thank three speakers, Dr. Yang, Dr. Yee, and Dr. Sughrue. Now it's our Q&A sessions. I'd like to remind everyone that you can submit questions using the Q&A windows below. Just let me share that. While we're waiting for more questions from the audience, we'll go ahead and launch 2 more poll questions. So first question is, to what extent are you utilizing external research platforms and services? Okay, looks like we have majority of people, the answers is all analysis and tools created by internal team. Okay, the second poll question is, what are the key barriers preventing you from publishing after grants and ethics are approved? Okay, while we're waiting for more questions from the audience, I have one question for Dr. Yang. Dr. Yang, now that you have a preliminary findings on the retrospective data using Infinitome, what is your plan in taking this project further, and what are the potential challenges?
Okay, so our next plan is try to work with more centers to acquire more multimodal data. So today we only show you the DTI results. Actually, we have also been actively collecting functional connectivity data using resting state fMRI. And sometimes we even use some active task fMRI. So if any people in the audience or people you know would like to join us, please welcome to contact the Infinitome team. So we can further compile our data further and to see what else we will together find. And also, I think the bigger challenge is that even though I have a lot of international collaborators, some of them are very concerned about the data sharing. So this actually is quite already solved by this platform, because the Infinitome platform have this like gap server shown by Dr. Michael, because in his slide he already explained to people that we will do this kind of de-identification task before uploading all the data to the cloud. And the data in the cloud is also saved in your country, your own region. So we are not sending data internationally. So through this platform, I think the biggest advantage is that people can rest assured that your data is not shared by any unlicensed party. It is just stored and in a third-party platform. And also the analysis is performed by a team of experts in that platform. Or you can also consult the team that how you can like do the analysis yourself using the special account they assign to you. So I think the next step certainly will be to compile more data and do a longitudinal study with more teams around the world.
Okay, thank you, Dr. Yen, and anyone who are interested in Alzheimer's study. I think we can work closely with Dr. Yen, and hopefully one day we can find a biomarker. I have one more question for Dr. Yee. Dr. Yee, we all know communication to glioma patients is very important before and after the surgery. How do you see the benefits of using this software in communicating to patients or patients' family?
Yeah, Jerry, definitely it's very helpful as well, because at least right now I have a very objective image that I can show to the patients and their family, by showing them, at least I can tell them that you see your tracts are all intact, your cortical areas are all intact. So most likely whatever you are seeing right now is definitely likely to be temporary. So unless I mean, it can go both ways, definitely. It can be used against, or it can be used to explain. But let's look at the positive side, whereby we can actually show the patient that we have done this safely, and we can achieve like I've said, all glioma surgeries, the aim is just to get a maximal safe functional resection. And definitely with the images, definitely it will help.
Okay, thank you. We are now 12 minutes over time. We are going to wrap this one up. There will be a short survey before you exit the webinar. Please kindly provide your feedback for our future improvements. If you have any more questions, please send us an email at contact@oat.com. On behalf of Omniscient, we would like to thank 3 speakers and everyone for participating tonight. Good night, everyone.
Good night.
Good night. Thank you.
Thank you so much. Bye-bye.